Welcome to the second video of the course on cell cycle. In this video, we will learn about the different cell cycle checkpoints. So in the previous video, we have learned about the different phases of cell cycle, and these phases must be carefully regulated and coordinated with both cell growth and DNA replication in order to ensure the formation of progeny cells containing intact genomes. In eukaryotic cells, the progression through cell cycle is controlled by a series of protein kinases that have been conserved from yeast to mammals. Defects in cell cycle regulation are the most common cause for the abnormal proliferation of cancer cells. So, the progression of cells through cell division is regulated by extracellular signals from environment as well as internal signals that monitor and coordinate the various processes taking place during different cell cycle phases. So, the control is accomplished by a series of checkpoints or control points that regulates progression of the cell through the various phases of the cell cycle. So, a major cell cycle regulatory point in many types of cells occurs in the late G1 stage, and it controls the progression from G1 to S phase. This regulatory point was first identified in budding yeast or Saccharomyces cerevisiae, where it is known as start. Once the cell have passed start, it is destined to complete one cell cycle. Now, this start is highly regulated event in yeast cell where it is controlled by different external factors like availability of nutrients, mating factors, and adequate cell size. The proliferation of most animal cell is similarly regulated in the G1 phase of the cell cycle. In particular, a decision point in late G1 is known as restriction point. So, in most animal cells, restriction point in late G1 functions just similar to start. However, the passage through start or the restriction point is regulated by external growth factors that signals cell proliferation in a contrast with start. So once it has passed the restriction point, the cell is committed to proceed to the S phase and the rest of the cell cycle. However, some cells they remain in G0 stage. So for some cells, they get arrested in this quiescent stage or G0 stage, which they can remain for long periods of time without proliferating. G0 cells are metabolically active, although they cease growth and have reduced the rate of protein synthesis. For example, in case of skin fibroblasts, they are arrested in G0 until stimulated by platelet derived growth factor to proliferate and repair wound damage. In some other cells, there are G2 checkpoints. So the fission is Saccharomyces pombe. The cell cycle is controlled by transition from G2 to M, the point at which cell size and nutrient availability is monitored. The control in G2 also occurs in animal oocyte. Vertebrate oocytes can remain arrested in G2 for long periods, like in human, it remains in G2 for decades. Progression to M phase is triggered by hormonal stimulation. So there are different checkpoints, and these checkpoints also check for DNA damage so that if there is DNA damage, it will not allow the cell to pass through the next stage. Out of these four stages, three are most important, that is G1 checkpoint or start or restriction point, G2 to M checkpoint, and spindle checkpoint, which checks the metaphase to anaphase transition. So, with this, we come to the end of the chapter, and we have learned about factors affecting cell cycle progression and different checkpoints of cell cycle. Thank you.